Yeah, hi everybody. I just wanted to. This is a supplemental recording, and hopefully it will come the way it's supposed to. Just want to make sure. Pardon me. This is what I was talking about in lecture, and it involves this particular chapter, which is effectively. Let me talk about nutrition and growth. This is very elementary physiology. It was one that had an extensive amount of notes called Chapter 4 notes, even though it's Chapter 6, it was Chapter 4 in another textbook, so I just happened to use it. But this is the growth. The other one's control of growth, which is a different chapter entirely. And this is highlights. Definition of essential. It has to be, the organism can't grow without it. So there are certain things that are essential. And here they talk about macronutrients and micronutrients. And then micronutrients, sometimes called trace elements, and usually these are things that you and I would call minerals: iron, zinc, as you can see here, nickel. Ours are typically things like iron and iodine. It's a little bit different for microorganisms. And macronutrients, basically, you're going to see the ones that are predominant very, very shortly. But the acronym to remember is CHON as in nitrogen, P as in phosphorus, S as in sulfur, CHOMPS, CHONPS. Those are the big, it's a little different in us. We have carbon, hydrogen, oxygen, and nitrogen, the big four, and then we have calcium and phosphorus. Here it's phosphorus and sulfur. That's a little bit different. That's the calcium is not, well, it's certainly an important relatively speaking, macronutrient, not the same. Bacteria, less so, prokaryotes, less so, microbes, less so. And so they're just all the different things that are there and where they're located. Don't care at all. Yeah, water. What's in there? Water, proteins, organic compounds, and here's that CHONPS. That's the one to take home. And usually and just like us, they ingest things and they break things down and we go from there. And I think that's really the important part. So hold on, I will get that in a second. I just got a text. Oh, it's my daughter at the wedding. Okay. So these are all the things that are about it. So only here's the CHONPS. They ingest it. They break things down. And so as you can see, we don't need a whole lot of nutrients, and neither do the bacteria. They can basically make everything. There are a few. And you're going to see the fancy name we give to them is called fastidious, Fastidious. What is meant by a fastidious bacteria is one that requires a lot of TLC, a lot of things that it can't get, and it has a lot of essential needs that are not easily or as easily obtainable. So I did it in my lab, and you'll do it later. If you don't have my lab, we used some blood agar. Well, the blood agar was pretty old, so it didn't work as well as it should have. But all aside, the essential nutrients were found to some extent, in red blood cells. So sometimes we use this in the laboratory, but that's significant. Most of the time, the organisms that we work with, particularly in the lab, are what are called facultative. FACULTATIVE. That means grow easily. Fastidious, difficult to grow. In general, as this course goes on, you're going to see facultative organisms, Staphylococcus, E. coli, pain in the neck to deal with because they grow so easily. They contaminate things. We get infections with them relatively easily because they grow so easily. They don't take a lot of help to grow. Whereas fastidious organisms, even though they can cause very, very troubling diseases like syphilis, Lyme disease, they need very, very special conditions. And those are things we can deal with. And if you did not know it, as long as we get to them quickly enough, they're eminently treatable. That's the highlight. So speed plays a role in this. So here you can see carbon, oxygen, nitrogen, hydrogen. You can see 92% right there. Now 96 phosphorus and sulfur. You can see calcium's way down there, but not so with us. It will replace It would probably be up here. It would probably, in our bodies, would replace with lower phosphorus and take its place up here, and sulfur would be by the wayside. That's just what I'm telling you. That's the only part to remember here is E. coli, classic. This is terminology I don't use. Okay. We are what are called heterotrophs. The bacteria we work with are heterotrophs. They basically eat a variety of things, extract them for building materials, carbon-based building materials, and do energy transformations. They break apart the same way we do. All those wonderful bonds, those covalent bonds with all the energy, use them as an energy source. So what we eat makes building blocks, and it makes energy. So it's very, very versatile. An autotroph is a plant or a photosynthetic bacteria. It uses carbon dioxide in the air, and it does what's called fixing carbon. It takes the carbon from the carbon dioxide and staples it together, affixing for FIX carbon, and that's what plants do in photosynthesis. And the energy they get is from sunlight. So they're photosynthetic organisms. They utilize energy that's available to them, make carbon-containing compounds and incorporate that energy, and they can use those carbon-containing compounds to make components inside the cell and tear it apart and utilize the energy if necessary. So they have not only mitochondria when we talk about a plant, but they also have chloroplasts. Photosynthetic bacteria, a little bit different, but they have sort of precursor chlorophyll, if you will, that are able to do that. Not a big deal, but just know the difference between heterotroph, many things to grow, autotroph, self-grow. So this is the typical autotroph, photosynthetic organisms such as algae, plants, and this somewhat exotic bacteria. There's some oddball things that are here. Rock eaters, ones that live on methane gas. Ugh. We are classically a chemoheterotroph, okay? And we get our energy sources, our carbon sources, and get energy, protozoa, fungi, many bacteria, animals. The other one is a little bit different from dead. That's when we talk about fungi or yeast. And certain kinds of bacteria are decomposers, breaking down the roadkill. And there's some oddball things that can utilize a live host. That's a big problem for us because they're gobbling us up, so to speak, in that process. That can be a big problem too. But in general, those are subcategories of chemoheterotrophs. Effectively, we are, if you think of us as men and women, as human individuals, organochemoheterotroph is the classic way it's described. Just that's that. Enough of that. No rock eating applied. See, carbon and energy from organic compounds. That's the bacteria we work with, etc., etc. Parasites, and by definition, we get to that a little bit in this chapter, and we reinforce it in the chapter that involves discussing, how shall I say it, interrelationships between organisms. Parasite, by definition, causes damage. Okay? No, I beg your pardon. A true parasite, as far as I am concerned, causes damage. Classically, we refer to this as a pathogen. A true parasite, whether it's living on the body or inside the cell, okay, and there's as you can see, a whole range of them. To me, if it's a parasite, what it's doing, okay, is it's thriving, and in some way or shape or form, the host, you, me, other plant and animal, are being damaged in some way, shape, or form. When they talk about ecto, as you can see on, endo, intracellular, we talked about this already a little bit when I introduced viruses. They are what is known as obligate intracellular parasites. They can't grow outside the host. They have to live inside the cell. Live is a wrong term. They have to occupy the inside of the cell and use its machinery. And as you can see, it's not just viruses. Here you can see leprosy and syphilis, both bacteria. So here, you can go through where the sources are. I don't do that, don't do that, don't do that, don't care. And on top of that, don't care. We're not going in, and this is a revisiting of something that you already should know. And I know it sounds like I'm racing because I am. You should know what diffusion is, and it's about concentration gradient. We know that. This is one way it gets in. Osmosis, selectively permeable, water, etc. And again, that whole thing about equalizing the concentration of water, we did this in AP1. And if you didn't have me for AP1, but most of you did, again, this is that whole idea of tonicity. So this is the osmosis, everything. Yeah, we actually used to do this experiment. I did this in high school. They have these osmometers, is what they call them. And you can see, depending on whether you make this if you make this hypotonic, it'll force the liquid in here, and the column will rise. Effectively, is what happens. This is the part that's important. This is a bacterial cell, let's say, with a cell wall. This would be like our cells without a cell wall. So if it's isotonic, the same solute to solvent concentration inside and outside of the cell, guess what? Nothing happens. You have as much water going in as going out. That's the desirable. Again, remember, this is an envelope. Now, if we're hypotonic, vast amount of, you have very little solute and a whole bunch of water, I think just pure water, deionized water. If it's a bacteria with a cell wall, it will stay in place just the way a plant does. It has mechanisms to pump out the water. Okay, that cell wall, it does nothing more than make sure the membrane against the cell wall holds it. You can't overwater it, even though some plants you can if you could overwhelm the mechanism. But the reality is in bacteria that doesn't happen. And it just will not take any more water in, but it will not destroy it. Whereas if it's one of our cells, it'll burst immediately. It's called plasmolysis, and that's rupturing of the cell membrane. On the other hand, this is why salt itself is a great preservative, because it pulls water out. And in bacteria, what does it do? It pulls the membrane away from the cell wall. Remember, it's an envelope that is going to burst. So then it's going to eventually it's going to cause plasmolysis here, as you can see, where that plasma membrane ruptures, and then effectively the bacteria will explode out. And even like in a red blood cell, remember the term is crenation, CRENATION. So there's the difference between cell walls and non-cell walls, and that's why even though it may not seem that way, it's a very similar phenomenon here and here, because it's lysing or bursting the plasma membrane. That's why highly salted water, years ago when we had a strep throat, the moms had this gargle with salty water. It was yuck, and ready to swallow and make it sick to your stomach. And they're reviewing passive and active transport, and you can read that. We did this. Microorganisms don't do a whole lot of active transport because they don't have a lot of energy resources. They generally ferment more than anything else. So there are some. Typically, these proteins and pumps are used to get rid of things in their environment that are harmful or bring things in that are necessary. Very, very limited. Like in antibiotics, you're going to see transport pumps when you see some of the resistance mechanisms that bacteria have, particularly gram-negatives in that periplasm. And it's because you have to utilize energy. And there's some things that are actively transported, as you can see. Don't remember them, but it's part of it. You don't have to worry about bacteria. Don't do this all because it's just about membrane-bound organelles. Some prokaryotic organisms do this. You can review it, but I'm not going to ask any. So when you talk about amoebas, they particularly can be a disease causer, but we're not really going to look at those mechanisms. So you can review it because you should. These are the areas I really want to emphasize. And then, of course, the bacterial growth curve. I may do the growth curve separately and just supplement you with that as well. But this is what's going on. Temperature. Why? Just like salting is efficient, temperature is very efficient. What makes temperature so efficient, okay, is living things, typically they're The temperature, what we call T optimum, where they grow best, is very close to T lethal. Our optimum temperature is 37 degrees Celsius. If you take your temperature up to 40, okay, at that point you're going to have 104 fever. You're really sick. If you take it up to 41, you're probably going to die. 42, certainly, you're going to be almost 108 degrees, 107.6. People rarely survive that. Even infants who are somewhat resistant to fever, you get to 104, 105, 103, you're in big trouble. Okay, critically ill. So that's the idea. The T optimum, it's not like temperature's not a bell curve. It doesn't go up and then go down. The typical bell curve, it goes up, and as you'll see, falling off a cliff. That's why temperature is so helpful. So these are the types of things that exist. And these are probably more archaea. These are the optimum temperature below 15 degrees Celsius. They can grow at 0 degrees, at freezing. Okay. If it gets any warmer, they can't. So refrigerator temperature causes them to grow rather than inhibiting. So the things that might thrive in the freezer section of your refrigerator, and they might, more often than not, we would see these. I don't know if you're going to see the ranges. So all of these polarized cap, deep ocean, typically we don't really. Psychrotrophs are interesting. Psychrophile, if you want to do the analysis of the name, phile means they really like it. Psychro means cold. Psychrotrophs grow in the cold, and they grow slowly. And you see they're somewhere in this range. So these are things that can grow in the refrigerator and cause foodborne illness. You have to be careful. Most refrigerators, probably around, if you want to think in terms, 4, 7, 10 degrees, depending on how you set your refrigerator, if you like colder or not. These are big players. I mean, a lot of food poisoning comes from toxins from Staphylococcus aureus, particularly with dairy that's been there too long. And this is we'll talk about Listeria monocytogenes. You read about it all the time. It can be extraordinarily bad for people who are immunocompromised, and particularly pregnant women. That is a real problem. And so we'll get into some of the things where we begin to see Listeria infections. So that you got to be so careful. Things like that. So that's in there. So understand that when you see the growth curve, when you chill something, it just generally slows the growth, particularly for these. Most of the things that affect us are mesophiles. They live in the middle. 20 to 40, 25, 20 to 45. There's a variety of ranges. Every textbook has one. In general, 20 to 45 is these, and you're going to see, and they overlap. It may be 40 to 70 or 45 to 70 is the next category, called thermophiles. They like it warmer. So as we go this way, these are the ones, animals and plants, soil and water. We're in a temperate, even in a tropical area, like where we're going shortly. And so typically the ones that this is where we are. We're 37 degrees Celsius. Some do pretty well, and these are what we see in heated or pasteurized food. So these are some of these endospore formers. There are certain steps you take. That's why when you can or jar something at home, if we do commercial canning, or if you like to can foods at home, effectively you're undergoing a very, very specific process. I'm not going to get into it, but these are endospore formers. These are thermophiles. Do they affect us? A little. They're better, hot springs, but these days we see them in hot tubs. Okay, so the range, like I said, is probably 45 to 80 or 70, but you can see we're not talking about the extreme or what we call hyperthermophiles. They're way above it. The ones to be familiar with for our purposes are psychrotrophs. They like cold. Mesophiles, the big player, because that's where all the things that affect us, and certainly these thermophiles. And this is what I mean. It's like falling off. I see none of these are bell curves. That's a classic example of these hyperthermophiles, the extremes. Boom, here. But let's look at us. Here's 37 degrees Celsius. Our T optimum is right about here. And then what happens when we get above 40? It's like falling off a cliff. That's why heat is so effective at dealing with why we use heat extensively for sterilization. It's cheap and it reliably kills. That's the beauty of it. You're going to learn it. We talked about in AP1. It irreversibly denatures things. So be on top of that. So you can see there's an overlap. A lot of authors will say 20 to 45 here, 45 to 70 or 80. And if you look at 10 textbooks, they have a similar chart and they all have different numbers. We have gases that are there, oxygen and carbon dioxide, and oxygen is a big player. And the trouble is toxic derivative of oxygen. This is really what we visit this a couple of times in a variety of courses. Any of my AP2, we're going to talk about toxic derivatives of oxygen. So, like us, we use oxygen and we can deal with these toxic derivatives because effectively what's oxygen is called the terminal electron acceptor. All it does its whole life is being bombarded with electrons. Boom, 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 boom. And so we have, as you're going to see, two enzymes in our peroxisomes. And you'll see the name shortly, superoxide dismutase, to deal with the superoxide radical, and catalase, something we're going to use in the lab, which is hydrogen peroxide. Okay, and catalase, basically, you'll kind of see the way it works. The catalase is there to detoxify hydrogen peroxide and make it oxygen gas, and water. Which is why when you put peroxide in a wound, because your cells have released their catalase, it bubbles and it stings because it happens pretty rapidly. And some can't use it at all. And you can see there's limitations. You'll see the categories. These are the toxic forms. Okay. This is the one that impacts our cells, the superoxide, singlet oxygen. And again, we use some of these things in efforts for sterilization. Here's hydrogen peroxide. Okay, and we can use it as disinfectant. We can even use it as a sterile. You can see it's very versatile. And then this is all of these are the reactive things that we get. And interestingly, we take this, the common one that affects our cells, change it to this, less toxic, and eventually use this, as I said before, to make oxygen gas. So here they are, two-step process. Radical, plus some hydrogen protons. We have them all around all the time. This is the enzyme that's in peroxisome, superoxide, and bacteria have some of this too. Not all. That's the big difference. Superoxide dismutase creates hydrogen peroxide and oxygen. Then the catalase enzyme takes the hydrogen peroxide, water, and oxygen gas. That's what effectively, when you put hydrogen peroxide on a wound, the catalase is exposed because tissue's been damaged, and it does this. Effectively, what happened, that hydrogen peroxide releases a lot of oxygen that destroy a lot of bacteria, and in the process stings, and when it evaporates very, very cleanly because you have water and oxygen gas. That's why hydrogen peroxide, as you will see, is used a lot in green products in the sense that they're environmentally friendly. So remember these two names, superoxide dismutase and catalase. And so what are we? An aerobe, a strict aerobe. We use oxygen. We have the enzymes. We call an organism that can't grow without it, like us, like a true aerobic bacteria. As you can see, fungi, protozoan, tuberculosis, got to have air. Obligate means you must have it to survive. And what they do is you do these tubes, and basically you can see when the tubes, and I've done this in labs, we don't do it in this lab, we inoculate it with different organisms, see where the growth is over time. We have to use a special media under special conditions. And what you see here is you'll see all the growth at the top. The one you'll see a lot of is Bacillus subtilis. You'll see if you grow that in a tube, you're going to have some stuff on the top of the tube. It tells you it's an aerobe. You'll see that in lab. Whereas microaerophiles, there's too much oxygen. Our atmosphere is 20% oxygen. They need somewhere between 2 and 10%. So that's either half to maybe 1/10 as much oxygen. So how do they grow? They grow below the tube. They have to have, some oxygen will get there, but not a lot. This is what you're looking at here is Helicobacter pylori. This is one that certainly plays a role in gastric cancers. It's found a lot in the stomach. It doesn't. People used to erroneously think it caused ulcers. It does not. I had major ulcers I've never tested positive, and that even today without the ulcers. And Borrelia burgdorferi, interesting, that's Lyme disease. This is what you work with a lot in the lab. Gram-negative internal, E. coli, Proteus, Enterobacter, Staphylococci. They have the faculty or the capability to be anaerobes. They do not require oxygen, but they'll use it. So they can ferment anaerobically, and if there's oxygen, they can function aerobically. Basically, they have a certain amount of catalase and superoxide dismutase. Obligate aerobes have an adequate, have a lot of these two, superoxide dismutase and catalase. Here they have limited amounts. So plenty, not enough. That's why they can only tolerate a little bit of oxygen. Here, they've got it. But they don't necessarily have to use it. Anaerobes don't have them. Okay, the intestinal bacteria, they are, not all of them, E. coli, but C. diff would be in this category. Oral bacteria, because there's an anaerobic area between dental tartar and your teeth. They lack the enzymes for dealing with any of it, and they die. That's why we use oxygen. You're going to see people have gas gangrene. That's a Clostridial bacteria. We put them in typically in hyperbaric oxygen, high area oxygen. We'll talk about that in wounds. That's really important. And you see, they all grow at the bottom. And these are interesting. And really probably the most interesting is some of the streptococci. There are some Clostridial species that are in there. They're called aerotolerant. They don't use oxygen. They don't do energy transformations aerobically or anaerobically, but they're not harmed by that because they have some ways, they have enzymes that can break down those derivatives. They just don't use any pathways for energy dependency. Sometimes we have to use extra bits of CO2, and you're going to see we need to grow gonorrhea, Neisseria gonorrhea, and Neisseria meningitis. We have to use special media that have that. Strep pneumonia, we have to sometimes use special media. We enrich the media to be able to identify that. And you're probably not going to see brucellosis, it's nasty. It's there. And effectively, when it comes to pH, the organisms, the ones that live at very acidic, we call acidophiles, or very alkaline alkophiles, don't impact us. The majority of organisms that we deal with are called neutrophiles, not neutrophils, which is a white blood cell, but neutrophiles. They're typically between 6 and 8, or even some ones are between 5 and 8. So some of the, not that these are not important, but not for us so far. As you can see, Proteus is interesting because it can adapt. As you can see, it can create alkaline conditions to neutralize the urine, colonize and infect the urine. So Proteus is kind of an Oswald. So this is like Staphylococcus can live in a very salty environment. So Staph aureus, and that's the way we isolate. That's what we're going to see when we talk about selective media in lab. That plays a role. But neutrophiles are what it is. I'm going to stop it here in the interest of time. I hope it's a good little nugget. And I'll give you the rest of this chapter in a separate dictation. And that'll be the supplements for when I'm away. Thank you.